Dr. Foat earned his undergraduate degree with highest distinction in genetics from the University of Wisconsin-Madison and his Ph.D. with distinction in biological sciences from Columbia University in New York City, where he was both a Sedgwick Graduate Fellow and Newberry Graduate Fellow in the Department of Biological Sciences. He is currently the Data Analytics Team Lead for Technology, Biotechnology, Trait Design, and Science, Genomics, and Data Science with Monsanto Company. From 2007 to 2009, Dr. Foat was a Genetics Fellow with the Department of Genetics at Washington University School of Medicine and awarded a postdoctoral fellowship in informatics from the PHRMA Foundation in Washington, D.C. He is passionate about building data analytics teams and driving the cycle of data-driven insight and real-world application via rapid synthesis of knowledge, methods, and computational technologies. And he is here with us tonight to talk about his experiences working to help farmers keep the world fed for another century and why he feeds his kids GMOs. On behalf of the Academy of Science St. Louis and Kirkwood High School, won't you please join me in welcoming Dr. Barrett Foat.
Thank you. Thanks for having me. Thanks, Rose, for that introduction. I didn't know I was going to give a whole CD ahead of my talk. So this talk was actually originally prepared for a platform address at the Ethical Society, where my family and I are members. And so that's why I don't have any slides. I will put up one visual aid. My talk is mostly about me and how I got to where I am, because I imagine as students thinking about where you might want to end up one day in your careers, you'll find it more interesting to see what is the career track that the scientist follows, which I think is how I got invited to give this talk. But I imagine some of you here also have questions about GMOs, and so I put up this website that I encourage you to go visit if you have more questions about GMOs, called gmoanswers.com. It's a website that is sponsored by all of the major companies in agricultural biotechnology, and you can actually enter your own questions there and get them answered by experts, people who probably are much more current with those questions than I am. You can also contact GMO Answers on Twitter, just say @GMO Answers. Okay, so let's see. No approvals were actually necessary for me to talk to you, even though I asked. All of this content is my own. I organized it myself. I've of course borrowed from a few different sources. Monsanto, as a company, values our engagement with the community enough that it's perfectly acceptable for me to come talk to you about my experiences and my knowledge and, as an expert in biotech and genomics. So I actually originally gave this talk, like I said, for the Ethical Society, and as I was imagining how I was going to give this talk, I imagined it could be the shortest possible platform address if I just said, I'm a biologist, I'm also a dad, and I feed GMOs to my kids, and I love my kids very much. And so if all of that is logically consistent, then everybody should feel pretty comfortable about GMOs. But of course, this is a scientific forum, and the Ethical Society is largely populated by people who value empiricism and rationalism, so you shouldn't just accept what I say because I say it. So of course, I have the rest of the talk that I wanted to give you. So I'm going to introduce it kind of with a story. I was back in New York City where I had done my graduate work. I was back there a few years ago in Manhattan for a scientific conference. And after a day full of scientific talks, I went to my favorite local pub to get some dinner. And it happened to be trivia night. So I sat at the bar, and there was this nice older couple that were sitting next to me who asked me if I wanted to be on their trivia team. And so we played a couple of rounds of trivia. I am terrible at trivia. My knowledge is very deep and not broad, but we had fun nonetheless. And so after a few rounds of trivia, we got to talking more about who are you and where'd you come from. And so the woman asked me, so what do you do? What do you do for a living? I said, well, I'm a scientist. Oh yeah, where do you work? Monsanto. Oh. But I thought you seemed like such a nice person. Well, actually, I am a nice person. And so that kind of gives you a feel for some of what I'm going to be talking about. This actually isn't meant to be a persuasive talk. I'm going to be talking about my experience and where I came from, how I got to where I am, where do scientists come from, as well as some of the interesting things I've learned about GMOs while working at Monsanto, a company that played a major role in the commercialization of biotech crops. And so, because this audience is largely students, you'll find it somewhat interesting to see the interplay between history and technology and my career. And so hopefully it will give you some insights into what kinds of things you could be looking for in current technology that might suggest opportunities for where you can go with your careers. So I was a senior in high school in 1997, 98, when the first bacterial genome, E. coli, was sequenced. And I had the smallest glimmer of insight at the time, realizing that genomics is really just getting off the ground. It's probably going to be a big deal. And actually, I just realized as I started talking about genomics that I need to give you all kind of a 5-minute version of Biology 101 if you're going to follow the rest of my talk. It's actually very, very difficult for me to talk about my life and my career without at least a little bit of basic jargon about biology. Okay, so I'm going to give you Biology 101 in 5 minutes. So there's a central organizing principle in biology. If you've taken biology here in high school, you've probably learned this already. The central dogma, so DNA, also known as the genetic material or the genome, codes for RNA, which is the transcript that gets translated into proteins. And so the combination of RNA molecules and protein molecules form most of the molecular machinery that make up cells. And of course, cells make up tissues, tissues make up organs, organs make up organisms, and organisms interact to form ecosystems. And so that's biology in a nutshell. I just saved you one semester in college. So I entered college after seeing the first genome sequenced, pursuing an undergraduate degree in genetics. And I went to the University of Wisconsin in Madison because that's where my family always went, all the way back to my great-grandfather. I happened to be really lucky that I that I actually went to the University of Wisconsin-Madison, that it was such a good school. I didn't go there for the reasons I should have gone there, but the University of Wisconsin has fantastic graduate research programs in all kinds of areas of biology. And so I ended up going to a premier research institution as an undergraduate. I happened to meet my now wife while living in the nerd dorm at the University of Wisconsin. And she said she was going to graduate in 3 years, and so I said I was going to graduate in 3 years, and that actually worked. And we both ended up moving together to New York City to go to graduate school, where I attended Columbia University in the Department of Biological Sciences. When I entered graduate school, I actually thought that I was going to go into neuroscience because that was something that Columbia University had special expertise in. I actually did 2 neuroscience rotations. And I was pretty sure I was going to join the lab of Marty Chalfie, who, after I had done the rotation in the lab, ended up getting the Nobel Prize as one of the co-inventors of green fluorescent protein. But as my last rotation, I just sort of on a whim decided, hey, I want to try computational biology, because we just had, the department had just hired a new faculty member in computational biology, Harman Bussemacher. And he was perhaps naive enough to take me on in the lab, me with no background in programming, and a fairly weak undergraduate background in math, which was common at the time, and I hope has been remedied in undergraduate education by now. And so I did my research in graduate school on gene expression and modeling and predicting gene expression, which really means learning how to interpret DNA, how to interpret the genome, to better predict how do cells determine which genes turn on and off when and why. I actually continued that research on my postdoc here at Washington University in St. Louis at the Center for Genome Sciences and the Department of Genetics, working with Gary Stormo. And I was applying for faculty positions in the fall of 2008 when the market crashed. And so here I am, my wife and I had our first child when we were graduate students, our second child while I was a postdoc, and I had a family of 4 that I was supporting on a postdoc salary. And the market just crashed, so we were in the worst economic period since the Great Depression. And at least put pieces together. Okay, so the economy supports taxes, supports the government, supports grants, supports research. This is bad. And it's probably not going to get better anytime soon. And so I gave some thought to, who else might have use for somebody like me? So at this point I was pretty good at programming, and I was pretty good at statistics, and I was an expert in modeling and predicting how genes get expressed in cells. And it just so happened that there was a job posting at Monsanto, actually not in St. Louis, at a remote site in Middleton, Wisconsin, right outside of Madison, which I loved. And I read the description, and it was almost exactly the kind of research I was planning on doing as a professor. It was like my research program I was applying for faculty positions. The only thing that was different was which organism. It was crops instead of, I was proposing yeast. And I read the description, and I was like, there's only like half a dozen people in the world who can do this job, and I know the other 5 aren't looking. And so I interviewed for that position and did well. So I joined a team called Gene Expression Technology. And Gene Expression Technology, the whole function is responsible for identifying parts of genes to put in and around the transgenes that we put into crop plants to get the biotech traits expressed in the right place, in the right time, in the right quantity in crop plants. So I did that for a couple of years, and then I moved back here to St. Louis to join what is now called Genomics and Data Science, where we do the bulk of Monsanto's discovery genomics. So genomics is the area of biology involving sequencing all of the DNA in a particular organism and interpreting that DNA to the extent that we can. And of course, because a genome is a decent amount of data, there's 2 billion bases in 2 billion bases in the genome of a human, and actually a similar number in a corn plant. So it's 2 gigabytes of data that codes for all of the information that makes a person or a corn plant. And so obviously you can't really look at 2 gigabytes and make sense of it, so that's where the computers and statistics comes in. And so genomics and data science, we are involved in analyzing all of the sequencing data that we produce at Monsanto for all kinds of genomes. Because we're involved in all aspects of agriculture, we sequence lots of genomes that nobody else really cares about, like weed species and insect species and all kinds of microbes. So some of the things that the team I'm working on now does include actually identifying new biotech trait genes, especially for insect control traits. And I'll talk more about that in a little bit. We also, when we put traits into crop plants, we need to make sure that they go in as intended. And so we also do analyses where we look at sequences coming of the plants that we put new traits into and identify and make sure that exactly the gene that we wanted to go in went in, nothing else went in, and it went in a way that we find acceptable and won't have any other effects. We also analyze a lot of data that's where we're sequencing RNA instead of DNA. If you sequence the RNA instead of the DNA, you can learn how plants are responding to their environments or how they're responding to a new biotech trait. And so we analyze RNA sequencing data. We also do analyses with genomes that isn't related to biotech traits. Monsanto has a very large conventional breeding program. And so we sequence genomes to help in the development of what are called markers, where we can trace natural genes that occur in crop genomes so that we can make sure that the desirable genes end up in the hybrids that we end up selling to farmers. Finally, one of the more interesting projects that our group is working on lately is effectively probiotics for plants. How many of you in the room eat probiotic yogurt or take probiotic pills? They're fairly popular now for improving aspects of digestion. crops have many associated microbes too. And so Monsanto has a large publicly announced collaboration for identifying are there any microbes that we can find associated with crop plants that improve their yield or tolerance to stress. Okay, so that's some of the work I do now and how I got there. So one of the things that I most actually enjoy about working at Monsanto, and a story that I don't think gets told very often in the GMO discussions, is very few people who talk about GMOs actually do it for a living. So it's actually kind of interesting. I work in an organization where everybody is in some way involved in the creation of biotech crops. And so I don't think the story of who these people are and what they're like really gets told very often. So the people who I work with in the Biotechnology organization come from diverse backgrounds and from all over the world. It's really, in a way, it's a cosmopolitan community. People from all different backgrounds, all different lifestyles, all different parts of the world, all working together in a hyper-collaborative environment. When Monsanto moves into new areas, it has a tendency to hire the very best experts. Because it's such a, whenever we're moving into a new area of science, it's such an important strategic shift that it's an exciting place to work as an expert because you can get resources to do research at a pace and at a scale that you could almost never do in an academic environment. But it's an interesting to watch scientists as they join the company. First, because we hire such fantastic experts, everyone we hire seems to have this really intense pride in being a scientist first. It makes sense. You spent 15 years trying to become a scientist, it's kind of important to you. And so there's this sort of deep value on doing really excellent science, having evidence-driven decisions, making sure that we're rigorous in how we're doing the science that we do. It's also interesting to watch, though, how people adjust to working in a research organization with the speed and resources that we have available when we have a major strategic initiative. Because we can do science so much faster and at such a large scale, and because it's also really, really collaborative, that everybody's on the same team, we're all trying to accomplish the same goal. And so, at first you see people come in, they have a sort of period of denial, where they still think their job is going to be like it was going to be if they were a professor, and it's really not like that at all. And then they realize how much more fun it is to do research when you don't have to be applying for grants all the time, and you get to do it at a large scale and at a high speed, and everybody's trying to help you. And you get to go home on the weekends and enjoy your weekends too. And so there's sort of this guilt that, like, wow, so doing science here is actually a lot more fun than I thought it was going to be when I was a professor. And then, of course, there's finally acceptance, and they get used to doing research at this hyper-collaborative research organization. So beyond pride in doing science, Monsanto's research organization has a great pride in developing its people, especially as leaders, which I have benefited from greatly personally. And there's also this real passion for the work we do. I never get up in the morning and think, why am I doing what I'm doing? Nothing that I do matters. I know that the research that we do at Monsanto has a real positive impact on the world, and you can feel that in the research organization. Everybody really, really believes that what they're doing is good for the world. So, I'm describing a little bit about the environment. We call both the organizational structure as well as kind of the culture, we call it the matrix, which has nothing to do with Keanu Reeves. The matrix is hyper-collaborative, which makes it a really fantastic place to do science. I was a little bit jarring when I first joined the organization on a postdoctoral fellowship. In a postdoctoral fellowship, at least where I did it, whenever you asked somebody for help, it felt kind of like a zero-sum game. That, like, if they were helping you, they kind of expected you to help them in the future. I had the experience when I started at Monsanto and I started asking people for help that everybody wanted to help me, like way more than I was even asking for. And it kind of freaked me out a little bit. It's like, why are you helping me? What do you want from me? But it's actually really just that collaborative. Another interesting side effect of this highly interconnected collaborative environment is that I analyze a lot of data for my work, and it was really baffling to me how I was going to find the data I was trying to analyze when I first started at the company. And I quickly found that the people network was vastly more efficient at finding data than the IT network was, which was a little bit surprising. That while I could never find the data I was looking for using the intranet searches, I only needed to ask like 2 people to find any data I was interested in. Also interestingly, because the organization is so highly connected, it's really bad at keeping secrets. When people are applying for new roles or their team reorganizations, like a huge swath of the organization knows about it before it's ever officially announced. And so, in some ways, like I've heard people have conspiracy theories about Monsanto, and I find that extremely hard to believe because I know all kinds of things that I shouldn't officially know, and there's nothing scary anywhere in there. Okay, so that's the environment, right? What it's like working at Monsanto. And I'm going to transition to the next part with a joke. You can obviously tell I love the people I work with, but this pokes fun at me and us in a meaningful way. Okay, so how do you tell the difference between an introverted scientist and an extroverted scientist? An introverted scientist stares at his shoes when he is talking to you, and an extroverted scientist stares at your shoes. So now imagine an entire research organization made up of such individuals who played a significant role in developing agricultural biotechnology. And they need to tell the world about it. How do you think that's going to go? Well, you know how that went. It went badly. And so I think that there's a tendency for scientists to not want to do what I'm doing right now and really put our face out there in the public and participate in the conversation. We kind of feel like, being sort of rational, empirically driven people, that, well, the data is good, all the data is out there, all of it's public, people should just look and they'll see that it's fine, and there's nothing to be concerned about. But of course, the world doesn't really think like scientists, and so it's really important for scientists, no matter what we're doing, to get out and talk about what we do and what we believe and why it matters. So agriculture is a technology, and starting before recorded history, humans have changed the genetic properties of the plants and animals that we've associated with. And we've done this by choosing the best examples from each generation and using them as the parents for the next generations. And this is usually called breeding. Through continued advances in farm equipment and agricultural practices, the amount of effort that's required to make a unit of food and the number of people who need to be making food has decreased to the smallest level of inputs ever in the history of the world, which is fantastic because that means that all of the rest of us don't need to worry about how we're going to make or find food today. We can do other things like science and art and build civilization and participate in markets. And do all the other things that make civilization work. So biotechnology is really only the last thin layer on a long, very thousands of years history of agricultural innovation. And with an expected world population in 2050 of 9 billion people, farmers are going to need to produce as much food in the next 50 years as they had in all of recorded human history. So the challenge is great, and we're never going to get there with those population projections without continued agricultural innovation. All world-changing technology faces resistance when it first arises. Just think of things like electricity or automobiles or air travel. But eventually society realizes the benefits it provides, and eventually it becomes such a normal part of our everyday lives that it doesn't really even worry us anymore. And I think that the promise for agricultural biotechnology is similar. That we're only just scratching the surface of the amazing things that we could do with biotech. So it's actually interesting to look at, sort of contrast the changes that have happened to crop plants by conventional breeding versus the small number of modifications we've done with transgenic biotech GMOs. It actually took scientists a while to even identify what the natural wild version of corn was, and it was eventually found in south-central Mexico. It's called teosinte, and it's this big bushy grass. You actually can't, without looking at it kind of sideways, you wouldn't really see how that turned into corn, because it has branches everywhere, and you don't see cobs anywhere. It's kind of straggly, and it's the transformation from this bushy grass to modern field corn is spectacular. Obviously, field corn, you only see a single stalk. It has these giant ears on it. And teosinte has these small seed pods with like 6 to 10 seeds, and they're covered by this hard shell. And you actually have to break open the shell to see what inside looks like a corn kernel. So breeding experiments have been done with you can actually still cross teosinte with modern corn, and you get all of these intermediate varieties that maybe suggest what it was like breeding teosinte into modern corn. You actually can see that this hard outer shell that's outside every kernel kind of got pulled back and turned inside out and became the cob. And then, of course, you got more kernels, and more rows of kernels, and a leafy covering. And so a series of small changes by breeding led to this dramatic difference in the yield of teosinte turning into modern corn. And so if you think about all the hundreds of genes that changed as teosinte got transformed by conventional breeding into modern corn, biotech is actually a pretty small change. It's just at most 8 genes on top of those hundreds of genes that have been changed in the course of conventional agriculture. Okay, so that's just breeding. What is a genetically modified organism? A GMO usually refers to inserting a gene that you can't do by breeding alone. That would never happen by breeding. Still, the genes that have been put into crop plants are natural genes. They're just not from corn or soy or cotton or whatever the organism is that we put in the biotech traits. It's actually also interesting that the mechanism that we use for inserting genes into the crop plants is also a natural mechanism that we've co-opted. There's a bacteria called Agrobacterium tumefaciens. How many of you have ever gone for a walk in the woods and seen trees with these like gnarly knobs on them, on the trunk or on the branches? Those are caused by a bacterium called Agrobacterium tumefaciens, called agro for short. What agro does is it has the ability to bind to the surface of a plant cell, create a microscopic tube into that plant cell, and insert some of its own DNA into the neighboring plant cell. And so a bunch of these cells will do that to the surrounding cells, and what that effectively does is change the behavior of those plant cells to create an agrobacterium dormitory with an all-you-can-eat free buffet. So what we've done is taken the agrobacterium, which is much easier to change genetically in a lab than any plant is. We removed the genes from agrobacterium that cause it to make the agrobacterium dorm, and we replaced it by the genes that we want to put into plants. And we use the agrobacterium itself to transform crop plants. It's also interesting to contrast breeding versus genetically modified traits. Breeding traits are always sort of random events that just by accident ended up doing something that we like, that we keep by breeding them into the next generation. All genetically modified traits were purposeful. There was a very specific gene that was intended to have a very specific purpose, and it went in as intended. So, kind of unlike breeding traits, genetically modified traits are engineered. So, genetically modified traits come in 3 major varieties for crops. There's virus resistance, insect resistance, and herbicide resistance. And I'll go through just sort of an example of each, because I actually think that once you kind of have a feel for how each of these work, they're actually kind of they're mostly cool. They're really elegant solutions to real agricultural problems. So virus resistance, viruses themselves insert genes into the cells that they infect. to make more viruses. And so it's actually possible to make an antivirus, like the antigene, that you can put in to a crop plant. That when the virus inserts its DNA or its RNA into the cells it's trying to infect, it will get bound by the antivirus RNA that's already in the cell, and that triggers the cell to degrade it, and so it can't have the infection. So insect control traits are actually using another natural mechanism. Have any of you in the room used organic pesticides on your gardens? One of the more popular ones is called BT, which is short for Bacillus thuringiensis. BT is actually an insect pest. So the reason it works as an organic pesticide is that you can apply it, you apply the actual bacteria spores themselves to your vegetables, and so when the insects eat a little bit of vegetables and get some of the bacteria in their system, then they'll get sick, and obviously then they can't eat your vegetables. And so the insect control traits are actually genes from these BT bacteria and similar species that the BT itself uses to get into the insects that it prefers. And it's actually interesting that there are many different strains of BT, and they have genes that target different varieties of insects. And so there's kind of this lock and key recognition mechanism. A specific strain of BT recognizes a specific kind of insect. And so the way the insect control traits work is we've identified these very specific genes for the insects that are crop plants. And we've taken them from BT and inserted them into, say, corn or soy instead. And so it's actually the crop itself that's expressing the BT proteins. It's sort of a natural pesticide that's already in the crop. And so what this results in is a whole lot less chemical pesticide needs to be applied. I think for sweet corn it's like a third less pesticide. And so if you think about the cost benefit there, that the BT protein doesn't bind to any of your proteins, your body digests it just like any other protein. And you can compare that to a chemical pesticide. So herbicide resistant, probably the best story is Roundup. Roundup is the name brand of herbicide called glyphosate. And glyphosate inhibits a particular step in the creation of one of the 20 amino acids. So animals, like you or me, need to eat, I believe it's 9 of the 20 amino acids. We can make the other 11 from sugars. Plants, obviously, because they're not eating anything, need to make all 20. And so glyphosate binds to an enzyme that does a particular step in the creation of just one amino acid. Because the plants can't make that one amino acid, it can't produce all the proteins it needs, and it can't grow. And so the actual herbicide resistance trait for Roundup, it was identified from bacteria again. The bacterial version of the same enzyme isn't affected by glyphosate, it doesn't bound. And so you can take the bacterial version and put it into a crop plant, and now that crop plant can still make that amino acid because it uses a bacterial version instead of its own, even when glyphosate is applied to the crop plant. So that means you can spray the entire field with the herbicide, and all the weeds die because they don't have the resistant gene, but all the crop plants are fine. This has had an interesting effect on farming practice. It gave rise to what's called no-till farming. You could actually, because plowing itself, one of the reasons farmers would plow would be to turn seeds from weeds back under the soil so they could germinate to keep weeds down. But if you could just spray your entire field with herbicide, you could actually plant your field and spray with herbicide to keep the weeds down without needing to plow. So if you imagine how radically much less fuel gets used if you don't need to drag a plow through thousands of acres. There's actually significant fuel conservation in no-till farming, not to mention preventing runoff. Okay, so one other misconception in the GMO conversation is how many things are actually genetically modified. There's actually relatively few. The major ones are corn, soy, canola, and cotton. And there are a few minor ones, alfalfa, potato, squash, papaya, sugar beets, and apples. And they actually kind of cracks me up around the grocery store sometimes because you see these labels on foods that say like GMO-free. And I look at the ingredients like there's nothing in here that's ever been genetically modified ever. So beyond the ones that are commercially available. There are some what could be kind of considered missed opportunities, sort of prototype genetically modified foods that haven't yet made it to market. So probably the best known is golden rice, which is rice that produces vitamin A. About 500,000 children go blind every year worldwide from vitamin A deficiency, and of those, about 1/2 will die from that deficiency within the year. Of course, rice is a major staple in some of the poorest areas of the world where most of these problems with nutrition are occurring. Golden rice was invented as a possible solution, such that if the rice was producing vitamin A, you could sort of alleviate this human suffering. The reason that golden rice is not yet available is actually very challenging to get a genetically modified crop through all the regulatory approvals worldwide that's required to commercialize it. And so my understanding is that's been sort of held up in the regulatory process for years. Other interesting genetically modified foods that haven't yet made it to market include allergy-free peanuts. Apparently the protein that causes most peanut allergies is required to make a good peanut. Chickens that cannot transmit bird flu. Aqua Bounty salmon that reach market weight using a fraction of the resources because they have another gene in them that causes them to grow much faster. It just happens to be a gene for another fish. EnviroPig, which produces less phosphorus in its manure, preventing algae blooms. This is one that might be close to home for this audience. Low acrylamide potatoes. When you fry potatoes to make potato chips or French fries, it often produces a compound called acrylamide, which is a carcinogen. Low acrylamide potatoes have modified the chemistry of the potatoes to prevent the creation of that carcinogen in the same quantities. Non-browning apples and fungicide-free strawberries are among others. So that's sort of GMOs and sort of why, some of the just stories about why I think they're actually cooler than they are concerning. And I think once you demystify the technology around GMOs and what they do, I think that makes GMOs much less scary than it just seems like scientists, scary scientists in a lab doing scary things. It's actually very nice scientists, often not in the lab, doing things that actually make a lot of sense. So I'll leave you with just one more story that kind of summarizes how I feel about working at Monsanto. Actually, the person in the story actually showed up in the audience today. I haven't seen him in months, but he showed up in the audience today to say hello and hear me talk. So I got to ask to use his name in person, because I actually hadn't seen him before I wrote this. A few months after I was hired, I was riding a car with my boss's boss, Greg, and his newly hired lead of sequencing, who I didn't get to talk to, so I'll call him Tim. And so Tim asked me, so Barrett, what brought you to Monsanto? Why did you decide to work here? And I said, well, I actually get to do the kind of research that I love to do, and I know that it can have a positive impact on the world. And Tim said, well, that's a pretty good answer. And he turned to Greg, who has been at Monsanto for a while and been involved in creating some of Monsanto's commercialized products, and said, so Greg, what does it feel like having had a positive impact on the world? And Greg, being somewhat quiet and humble person in general, kind of has a half mile and says, gets me up in the morning. So hopefully I've given you a feel for who are the kinds of scientists who do work on genetically modified organisms at Monsanto, where do they come from, what it's like working there, working with that technology. And I hope I've given you some sense of how that technology works to maybe demystify it and hopefully give you some of the cool factor that you don't usually get in the conversation. I'm going to give you a few resources here before I go into talking a little bit more about sort of career advice for students, which you might find pressing, seeing where you all are in life. I got some of the material for my talk, including the list of currently commercialized genetically modified crops, as well as missed opportunities, from a talk by Kevin Folta, a professor at the University of Florida. I put up earlier on the screen a website, gmoanswers.com, which I recommend you checking out if you have any other questions. There's also, at the end of the talk, there are pamphlets up at the end of both aisles for gmoanswers.com if you want more information. Monsanto also has a website that talks more about our research, discover.monsanto.com. Okay. So on to career advice. If you are interested in being a scientist, first make sure you really, really want to do it because you're going to be working at it for a very long time. Most people who are considered scientists have a Ph.D., sometimes they have an M.D. You can have a career in science without a Ph.D., but they tend to be more technical careers where you're acting under the supervision of somebody who is a Ph.D. scientist. One of the most important experiences to get if you have an interest in getting a career in science, whether it's only getting a degree in a bachelor's level or master's level degree or going all the way through to a PhD, is to get experience doing real research in a real lab as early as possible. And so it's actually pretty important to, when you're choosing schools to go to as an undergrad, to choose a school that has has abundant opportunities for undergraduate research, whether it's intentionally as a formalized program or just there's lots of graduate research that happens and there are lots of opportunities for internships and lab positions. I also have never met a scientist who was a bad student. So it's obviously very important to get good grades and to test well. And I think increasingly, and you'll hear more of this when I talk about sort of megatrends, you should learn how to program and you should take courses in statistics. And you'll see why in a second. So I think when I was thinking about advice to give to students who haven't yet started their path towards what their careers might be one day, I think the most useful information I could give that I had the intuition, which was lucky, but nobody ever told me this. It's not useful to think of a career as a track or a path. It's especially if you want to do a technology career, science or information technology or any technology career. You should think of it more like surfing. That the best way to have a technology career is to sit on your surfboard, which is sort of like your skill set, and look out at the horizon at the waves that are coming. And make sure that you get your board moving in the right direction, just in the right time, at the right speed, that you can get up at that wave when it arrives. And so it's actually useful in thinking about your career is like, what are the things that are just starting now that aren't even close to being sort of full-blown trends that will sort of arrive when you're graduating from college or graduating from a PhD program. So things I see happening now, one wave that is just really large, and so will probably still be going by the time you all get there, is what's called data science. Data science is actually a fairly broad term, which refers to the intersection between programming, statistics, and machine learning, and then some domain. And that domain could be anything that generates significant amounts of data. It could be a science, like biology or physics, or it could be something that is connected to a market, like the stock market or marketing, or social science. So any domain with statistics and programming. It's actually fairly difficult to be good at all three. That if you're expertise in a domain, also being fairly good at programming, and also being fairly good at statistics is fairly challenging. So people who can cover all of those bases will always be in demand because it's actually a very difficult thing to fit it all in my brain. When people come with this level of training, they're immensely valuable though, because if you think about how much faster somebody can move if they have the domain expertise to ask the right questions, and they have the programming and statistics skills to extract answers from that data, that's radically more efficient than if you had to assemble a team to do the same thing. So medical genomics is a trend that's been coming for a while, but still isn't here yet. So I expect easily within the next 20 years, if not the next sort of 10 to 15, we'll all have the ability to get our genome sequenced. And if you imagine what that means for medicine and the practice of medicine, that if you have everybody in the country has their genome sequenced and that information is consulted for what's your predisposition to diseases, what medications are you going to have positive or negative reactions to, maybe what allergies might you have that we don't know about yet, etc. When you have hundreds of millions of people who all need to be consuming that kind of information, the opportunities that creates for database creation and software and statistics and counselors to help patients understand that information and instructors who help doctors better communicate that information. There's going to be a giant industry that gets created from everybody's genomes and being able to consume that information for the practice of medicine. And so in order to capitalize on that trend, of course, you need a degree in biology, experience in software engineering, and statistics. You could also imagine rolling in sort of communication or user experience aspect of that, because I think a big challenge is going to be as much in communicating to doctors and patients as it is to doing the actual data analysis itself. So the third major megatrend that I see coming is the Internet of Things, which is another vague term like data science. So the Internet of Things refers to how devices are increasingly connected to the internet. Like all kinds of things are connected to the internet. And so if you think about the Internet of Things in the context of science, imagine what it would like to be doing any kind of science when sensors are so cheap they're almost disposable and they're connected to the internet. What would you do if you could spray sensors across the landscape? What would you measure? What kinds of analyses would you do? And so that's sort of more about blue sky, that the Internet of Things and its effect on science and how we understand our world is going to be amazing. Yeah. So just to reiterate, I think the best advice for a technology or science career is to aim where the science isn't yet. Where is it going to be when you get there? And finally, the last message, given the title of my talk, I feed GMOs to my kids. There's a lot of noise in the discussion about GMOs. And I don't know if any of you have participated in formal debates before, or maybe informal debates, but you should never interpret the volume or the frequency with which somebody expresses an opinion for its correctness. So thank you all for listening, and I understand the way it'll be.